that's sort of how I felt at that time, right? When you speak to people who have spent decades in this industry, it almost feels like, okay, where do I get started? Where can I be the most impactful in terms of my skill sets? And so I think just getting started and starting to even just self-learn, there's a ton of value in that. And obviously, taking classes at school or shadowing people who have been working in the space, I think is another big thing. I think you'd be shocked how often people are just willing to, chat and help you out. And so taking that first leap of faith and reaching out.
Welcome back to Discovery Engines, a podcast on the people and the platforms who are accelerating scientific discovery. I'm your host, Nabil Laoudji. The biotech, materials, and chemicals sectors hold a lot of potential for good, whether it's developing new cures for disease or new materials to power our energy-hungry planet. And yet, contributing to these fields can feel daunting. If you're in school, you might find yourself staring at a mountain of textbooks, competitive coursework, and long hours in the lab. If you're out of school, pivoting into these areas can feel like an even bigger mountain to climb. And there are good reasons for this. The science is intricate, and the responsibilities to create safe therapeutics and materials carry real impacts on society and the planet. But these barriers can also keep people out with innovative ideas and a strong sense of purpose. When the potential impact to humanity is so high, how might we lower the barriers to participation and attract talent that might otherwise be drawn to opportunities that are easier to enter, but less societally impactful? One person who hasn't let those barriers get in her way is Alishba Imran, an undergrad at UC Berkeley who early in her career has already contributed to multiple scientific endeavors. A deep learning researcher with Berkeley's BAIR lab, Alishba is also a research fellow at the ARC Institute and the co-founder of Voltx, a startup that built next-generation battery testing platforms. Alishba's completed machine learning internships at organizations such as Cruise and Tesla, NVIDIA, and the Chan Zuckerberg Biohub. And she's the co-author of the forthcoming book, AI for Robotics, Towards Embodied and General Intelligence in the Physical World. In my conversation with Alishba, we discussed how she bootstrapped her learning in biology and in chemistry, what it takes to launch a deep tech startup like Voltx, Alishba's work advancing cell biology with self-supervised learning, the promise and the hype of robotics research today, and how nonprofit research organizations like the ARC Institute are reshaping how science is done. If you're curious about the future of automation, biology, and AI, or just want to hear from somebody who has cleared their own path into contributing to AI for science, this episode is for you. Let's dive in. All right, Alishba, welcome to Discovery Engines.
Thanks for having me.
Thanks for being here. So we connected about a month ago on Twitter to talk about having you here on the podcast. I actually ran across your profile a few months before that. I was putting together this autonomous science research map and had run across some of the work that you've done at the intersection of AI and machine learning and science. And have seen that you have worked with some really cool organizations on some really interesting projects at that intersection, which is personally fascinating to me. And so excited to learn more about those experiences today and get your insights and perspectives.
Yeah, excited to chat.
Awesome. So maybe just starting off with just some quick grounding. So you're currently an undergrad at UC Berkeley. You're studying computer science and electrical engineering and math. As part of that experience, you're a deep learning researcher at Berkeley's BAIR AI research program. You have previously co-founded and were CTO of Voltx, a company in sort of battery technologies, and we'll get more into that in a minute. You've had some really cool internships in machine learning with organizations like Cruise, NVIDIA, Tesla, CZ Biohub. You're currently a research fellow at the ARC Institute, and you have a forthcoming book coming out calling AI for Robotics. Did I get that right?
Yes.
Okay, awesome. So lots to dig into. So maybe just starting off with, I think, the passion that we both share, which is around applying AI ML into the sciences. So we're in the Bay Area right now. When I look at the companies that are getting the big funding rounds and getting a lot of excitement, a lot of them are in things like coding assistants or new types of chatbots or video generation technology, and that stuff is really cool. That said, a lot of your experience seems to be really on the applications of AI and ML towards science specifically, so chemistry and biology in particular. Where did that interest come up for you?
Yeah, I think for me, I guess I started learning about AI pretty early on in high school, and it started more as just like a curiosity. I remember, I'm from Toronto, so there was a lot of cool stuff happening at the Vector Institute with Geoffrey Hinton and some really exciting just research going on, and I was generally very curious about sort of this paradigm of AI and ML, and at the time I had just learned and started coding and started just dabbling in it, and so just started going to local events, and got to see talks from people like Geoffrey Hinton and him mentioning just go home and just read a paper, right? Learn about backpropagation. He was describing his research. And so that got me really interested in just the field in general. And so I guess initially the curiosity started with more just generally what is AI and ML, started reading papers myself and taking courses online to teach myself about the sort of key methods and algorithms in the space. And then I would say towards the end of high school, I got really interested in sort of battery technology, and I would say the main curiosity started from more so just climate change and sustainability broadly, and energy storage is sort of one of the key aspects of that problem, as to why we don't have, if you look at the cost of renewables, for example, they're actually much cheaper than non-renewables, but the reason we're not able to reliably use that is because we don't have long duration energy storage. And one of the most common systems for that is batteries. And so me and my friend at the time were just really interested in this space. We both had a computational background, and so we started diving really deep into batteries and the supply chain, but also sort of the material science side of it, and the chemistry of how these batteries work, what are the degradation patterns and failure methods. And I would say that's where my curiosity for science really started. It was whoa, this is so cool. And, I discovered this newer field of material science and a lot of really cool research happening at the intersection of machine learning and material science. And so that's where I realized, there's a really cool application. That's partly how I started my company, which I can talk more about. But I would say that was sort of the initial spark of hey, there's some really cool science that can be accelerated through just computational tools. And then as I came to college, as I came to Berkeley, I have a ton of friends that are doing more biology research, and also, so I think just learning from them, and also, reading about things like AlphaFold and a lot of the stuff happening now in the protein modeling space, and obviously the Nobel Prize recently. I've just been really keeping up to date with that field, and I realize there's a ton of work that's happening in the biology space, whether that's applied to medicine or even sustainability, right? There's a lot of applications of using biomaterials, for example, for batteries, and so I think that intersection is another area that's really cool. But I would say broadly, there's just a lot of interesting problems, and also it feels like they're very applicable to the real world now, and so as these algorithms are getting better and we have more compute and data, how do we really apply these to these harder scientific problems? So that's sort of my journey in the field.
For folks who don't know, Geoffrey Hinton, often referred to as the godfather of AI, I think was in the early days, maybe it was at Google, coming up with some of the first technologies that I think underpin a lot of how large language models and AI models are built today. Going back to, I think, your interest in sort of the battery application, you mentioned climate change and sustainability, so I hear sort of a maybe more kind of purpose-led mission around that. Would you say that there is a similar path in biology, which we'll get to next, around purpose, or is it just, hey, the science here is really cool and there's some kind of cool buzz with things like AlphaFold happening?
I mean, definitely, right? I think at the end of the day, it's you want the biology research to be impactful, whether it's for developing therapeutics or eventually getting drugs to pharma, right? That's really the bottom line. And there's a lot of sort of intermediate work that can be done there in terms of building methods that sort of just narrow down the search space for, if you're finding a target for something and if you can build an algorithm to lower that search space, that in itself could be really useful. And so I think there's a lot of sort of stepping stone work that's happening now that hopefully eventually, can be useful for getting these drugs to market. But yeah, I think definitely in all the work I do, that's sort of the main thing I look for is like, can this work be impactful? Maybe not immediately, but will this set the foundation for maybe helping researchers in the space that can then, build on this to get a drug to market? And I think eventually that's what I would want to work on is like, hopefully, if it's in sustainability or biology, really getting something to market.
One of the hurdles, and my hope and intention is for this podcast to sort of lower the barriers to entry for working on these kinds of problems, is that, when you think about battery technology and chemistry and biology, these are like very technical field with deep science, and a lot of folks who work in that field, have really, labored for many hours in wet labs and, pipetting and growing cells, and it's just a really tough field to come up through. And there's an incredible amount of depth and complexity to it, seemingly infinite. Somebody recently described this, for example, a human cell as a black box full of a million, other black boxes inside of it. You have this really interesting background in math and kind of on the computational side, and yet you have, not just dabbled, but really, it seems like, dug in and leaned in to applications of, ML, AI for these spaces. How did you get up to speed? How did you overcome that barrier to find a way to add value?
Yeah, I think that's a really good question. And I think it can be definitely really scary as someone starting in the field to be like, where do I even get started, right? I think there's a ton of jargon that's used in the field for people that have spent decades just working on these things. But yeah, I'd say I did a combination of a few things. I mean, even this past summer, I actually started just like a reading group where I was reading a few textbooks that the people that I was working with at CZ Biohub had recommended, and I just started, they let me borrow their textbook, and I was just like spending time after work reading it, and it was a ton of fun. And so I just posted about it and like got a ton of interest from other people that were in a similar position. And so we started this like really informal reading group where it was just us sort of reading different papers or textbooks, and then just hopping on a call once in a while about, just chatting about what we learned and if we had any questions and just walking through it together. And I think that's a really good way to learn is, if you can synthesize a really technical topic and share it with someone else, I think that's a really good test of your own understanding. So one thing I'd say is definitely lean into like the textbooks and sort of like the more traditional, resources that are out there that do help you get a baseline understanding. Obviously, I think another thing is getting hands-on and working on projects. So I think the work that I did at CZ Biohub was really helpful and complementary as I was learning and reading those textbooks, because I was able to sort of see I was doing a ton of work with like single cells and really diving deep into the organelles and the dynamics of how these organelles are changing and shifting due to different perturbations. And so really understanding, okay, these are the organelles that I was just learning about, here's what they actually look like, and here's how they change over time. And so I think doing an applied project is really useful. And I'd say the third thing is like definitely, to practically really understand things, you do have to get into a wet lab and get your hands dirty with some of the tools. Even if, you're not the wet lab person, maybe you enjoy the computational work more, I think that's okay. But I do think there is a lot of value in actually learning the skills of being in a wet lab. So I've spent a ton of time just like shadowing, and also even, in the summer, that was something that I tried to get closer to with the people who are collecting the data and doing the experiments, just, even just chatting with them and understanding how do they go about designing these experiments and how does that tie into the downstream work of the algorithms that I'm building. So I think one, spending time shadowing people, being in the wet lab, but also chatting with the biologists and really understanding what they're working on.
That's great. Well, one is shout out to textbooks. I feel like, I went to college a long time ago, felt like textbooks were on their way out, but great to see that they still provide some value and can be kind of a cool resource for a study group. I also want to give a shout out to, in the Bay Area, there's an organization that I've been kind of helping out with. They're called Celsius. I think they're changing their name to Biopunk Hackers or something, but needless to say, they are essentially a community like wet lab space. They provide all sorts of training and access. I imagine that's not unique to the Bay Area, but there are some really cool folks, I think, who are trying to make this science and being able to go into a wet lab a lot more accessible for folks.
Yeah.
One of the things that I heard that even surprised me was that sometimes folks who even work in industry at these large companies, they themselves, maybe because of understandable protocols and so forth, aren't actually able to get access to wet labs, even if they want to, let's say if they work on the dry side. So I think folks who are willing to maybe take a bet and help somebody out or create these kinds of organizations are just really helpful for making that more accessible.
Yeah, I think there's a ton of like open spaces in SF definitely that have just like, space you can rent out. And but also I think, I have a friend who goes to like, he goes to UMich, and he was mentioning there's like a class you can take where you can just like learn about sort of like the fundamental skills in wet lab. And so he came from like a more neuroscience background, and so that really helped him just get up to speed with just like what are sort of like the main protocols and things to be aware of when you're like designing different experiments and collecting data. And so I think it really helps to have someone who has spent a lot of time doing that walk you through it. And I think you reach a point where things become a lot more clear and like repetitive, and then you're able to sort of apply those skills yourself.
Well, we have been talking about chemistry and materials and biology. So let's maybe go back to the chemistry and batteries, I should say. And so you had this company Voltx that you started, I think, while you were in high school. And it's described as a venture-backed startup focused on optimizing and accelerating the development of electrochemical devices. I think you said specifically the average test for a batch of battery or supercapacitor cells takes anywhere from 3 months to 1 year. And then developing the technology that you did, you're able to shorten that to a matter of days. So maybe just zooming out, why is being able to just test batches of batteries like a useful or important thing for the industry, and being able to do that more quickly?
Yeah, it's a super important process for any battery manufacturer, right? So if you're an OEM or just a manufacturer in general, it's something you have to do as part of one within R&D as you're testing. Like, let's say if you're looking at a new cathode or anode, you need to test to see, hey, is this going to be a good battery cell that's going to last, X amount of years for whatever application I'm looking at, or even within like, quality assurance and reliability, you need to say, hey, this battery cell will last, 10 years. And so there's sort of two different aspects of it. And testing is done throughout the entire pipeline of a battery development, right? And so I think it really becomes a huge bottleneck in terms of the throughput of these cells, and also just in terms of advancement and finding new battery cell chemistries, because essentially it's a super manual process, right? And so usually you have a battery cell, and you hook it up to this device that collects your data, and then you are essentially just measuring the capacity and the voltage and like the resistance increase over time in the cell, and that gives you a really good proxy for just like degradation and how long will this battery cell last. But really, what we realize is like these companies are collecting a ton of data from these experiments, but they're not doing anything with it. And I think that's where the key insight came in of, is there a way we can sort of use that early on data that they're collecting to then predict sort of the degradation of these cells such that we can save time on the battery tests. And obviously it's a really complicated problem. You can't just look at sort of like the output metrics of voltage. You need to really understand what's happening inside of the battery cell. So we looked a lot at physics-based simulations as well, and understanding the different mechanisms in which a cell usually dies, and how what's actually happening inside the cell essentially, to see if that would give us a better proxy for understanding the life of the battery cell.
So I imagine, one application of that is, hey, I'm a manufacturer making a ton of batteries. I want to make sure that the batteries are going to meet sort of what I have on the warranty or what I'm promising the customer, and that they're not going to degrade before that. And having a technology like yours is helpful for that. I imagine another sort of application could be, and maybe you alluded to this earlier, is, hey, what are some alternative chemistries that we could come up with here so that we can develop just batteries with consistently better properties long term? So maybe can you share more about sort of like how much of your focus was on the former versus the latter?
Yeah, we definitely started more with the quality assurance and reliability problem. So there usually you have a chemistry that you've already sort of tested and validated, and then you need to sort of mass manufacture these cells. But each batch, there can be different batch effects, and so you need to still test a few cells within each batch to give that sort of warranty. And so that can still be a huge process. And so when you think about I guess the value add very practically, you can measure sort of like time and cost saved as an effect of that. But a lot of the insights we gathered from that, we also looked at sort of R&D, because it's, you could use very similar sort of training methods and data, but it's obviously a harder problem because now you might have more variability in terms of the chemistry, and so you need to build that into the model as well. And so, yeah, I'd say we initially started with that and also looked into R&D a lot, as I do think that's it's a big area of development and impact as well.
I recently had the opportunity to tour the A Lab at Lawrence Berkeley National Lab, and they do some really cool work in automated materials discovery with a focus on battery technologies, and they are developing, they say, new chemistries such as disordered rock salt battery chemistries, which basically, as I understand it, have better power energy density, are safer. Batteries, I guess, are less prone to these kind of uncontrolled fires, which can happen with current lithium ion technologies. I think in speaking with a friend of mine who works for an electric airplane startup and is also working on batteries, he said, these new chemistries are really exciting and really cool, but there's also a lot of practical challenges that I have to think of as a manufacturer or a product creator, that I need to be able to get access to these materials at scale, for example, and scale up a manufacturing process and so forth. And so that then I think leads to folks being maybe a little bit more incremental in the kind of chemistries that they play with. I'm curious, what insights you might have had into that as you were sort of in the R&D side of the space.
Yeah, I think there's a ton of cool work. I think one area that's super impactful that's still pretty unexplored is just like raw earth minerals, right? And if you think about sort of like the amount of demand we're going to have for electric vehicles or just in general with like renewable sources, that's going to way surpass sort of our capacity of available raw earth minerals to build these batteries. And so I think one area that's really interesting, one thing we found was with a lot of companies we chatted with, they actually were starting to build out a recycling facility aside from their manufacturing facility, and that was solely focused on sort of how do we do like second-hand life testing for these batteries to see if they can still be used. Maybe we can extract some of the materials and still use it to build new battery cells. And so I think that's one area that's super exciting, and an area we were looking at because that's another area where there's a ton of extensive testing that's being done, and so you still need to understand the battery cell life to do that. And then another area is also just, are there other types of, like, how do we get these minerals and these materials maybe in like a synthetic way, or generate them in some other manner without needing to really like extract them from the earth. So I think that the area of like synthetic minerals or materials discovery is also really interesting, and I think will be really important. And so you bring up a good point, right? With these new technologies, people are finding new materials, and there's a ton of different chemistries that are being developed, and everyone is sort of saying, hey, our battery cell is the best. It's going to last the longest. It has the highest energy density. But I think really it comes down to how accessible are those materials, and that really will end up being the root cause. And so if we're able to solve that even for like lithium-ion batteries, I think that would be super impactful.
Yeah, it was, as we're talking more and more about sort of onshoring a lot of manufacturing, I saw some statistic that like, the average iPhone has, materials from like, 40 different countries. And, I think that raises the question of how do we then find those materials in a cost-effective way. It does seem like being able to, I don't know, maybe synthetically create them in some ways or so forth would be sort of like the golden ticket for being able to unlock a lot of this sort of like supply chain independence that I think we're looking for. Yeah. Cool. So tell me more about sort of the business side of this startup. So, there's a lot of scientists, founders out there might be listening to this podcast. They've got their own ideas for starting a business with some sort of technology or maybe algorithm that they have that they think could be useful. Like, what was your experience of trying to raise money, trying to create a sale, managing sort of the financial side of this business?
Yeah, I think there's a ton of learnings. I think obviously starting a company, there's a lot of your thinking really goes into just spending time with customers, right? And understanding what is the problem that they're willing to pay money for. And that's something, I was thinking about earlier today as well. I was like, if I want to, build something that can be really productized, what is really the bridge between, doing research and getting that into a market, and hopefully, for it to be impactful and useful. And I think sometimes you really need to approach the research problem from a perspective of, how do I make this useful? And so being really close to your customers, right? Whether that's like pharma, or in our case, it was, talking to battery manufacturers. When we initially started, we just did, a ton of outreach, just really reaching out to all the companies in the space that we knew of. And also just like leveraging our own contacts. We knew someone at Tesla, for example, and he was a VP there. And so he was super helpful in being like, hey, I'll connect you to, a few people that I know that might be interested in this. And so initially, we would go into the call not being like, hey, this is a problem. Here's our solution. What do you think? But instead, we'd be like, tell us, your top 3 pain points. Walk us through your entire value chain and like where are the biggest sort of inefficiencies that you see right now. And so really approaching it from like a problem discovery perspective rather than slapping on your solution and just asking for validation there. So that was the first thing we did. And I think that was really helpful in identifying a problem that people were willing to pay for. But also when you think about, pricing models and how much can you really charge them, you need to understand what is the value you're adding. And how much can you charge based on that? And so I think understanding the current value chain was really helpful in being able to do that and get that data to then price our models. But then also it was a great way to sort of build like an initial discovery board, I would say, of companies that were like, hey, this piques our interest. Like we might be, if you have a solution for this, like we would love to use it. And so we sort of had a list of companies that gave us that initial like spark. And what we would do is we, just using open source data that we found, we built sort of an initial model, got some results from that, and we would just send them updates every few weeks being like, hey, this is a result we got. What are your thoughts on this, right? And then eventually that led into, translating 1 or 2 of those companies into real pilots because they were very interested, and so they ended up giving us, data from their manufacturing facilities, and we'd use that to then like update the models and train them. And so I think that was a really helpful process, not approaching it really as like intentionally trying to sell to them, but really just understanding the problem. So that I would say is more sort of on, I guess, like the pricing side. And then raising money, I think, is really, a lot of it is just understanding how to sell what you're working on. And so I think part of that for us was making it really clear why we wanted to work on this problem. I think if you talk about sort of the mission-driven impact that you want to make, that really helps. I think a lot of VCs obviously want to see that to understand, are you really in this for the right reasons, and are you willing to stick through it even when things get hard? And so I think that was really useful. And then also, again, a lot of it is just knowing the right people and reaching out to people in the space. And so, just asking contacts that we had. Hey, do you know anyone at this firm, or people we can chat with? And again, just really approaching it with like we're building this thing, let us just keep you updated, and potentially, if we raise in the future, we might reach out again. And so I think that was really a key theme in all of this, was it was never really intentional. Like we wanted to start a company, or, we wanted to raise money. It was always like, it felt like the right thing. It was what we needed to do. And we had just, started building those relationships to get to a point where it made sense to do that.
I love that insight, really staying close to your potential customers and in a way almost co-creating with them by really understanding their needs and then prototyping that, sharing that back with them for feedback, not necessarily even trying to sell them something, but then ultimately that leading to a couple of maybe, access to interesting data sets that you might not otherwise have, and then actual clients for your product.
Yeah, and I think you have to understand that the sales cycles are really long, right? If you're working in any market that's where you're trying to really disrupt the way things are working, if whether that's like pharma or whether that's like, in our case, battery manufacturers, like it's just obviously there's a certain way that things have been done for ages. And if you're trying to really come in and say, hey, we're going to change the way things are done, it's really an iterative process. And so the sales cycles do end up being really long. But I think if you build that initial sort of relationship with them, it helps sort of shorten that period as well, because they start to trust you. They see that you're really diligent about working on the problem.
Well, in some ways, I feel like, gosh, a few years ago, everybody was talking about EVs. There was a lot of investment happening there. A lot of people were buying them. Then, it seemed that that's dampered down. People were saying, we're having trouble selling EVs. And yet there still seems to be excitement in the space. So I think just a couple of weeks ago, BYD, one of the largest EV manufacturers in the world, had this glitzy PR release about how they have a new battery that can be charged in 5 minutes that can get you to, 80% or 100% capacity, almost equivalent to filling up your car with gas. And that was really exciting. And so there still seems to be a lot of investment in R&D in this space. Like, what do you make of sort of the state of battery tech today? Is this still a space that you would encourage folks to, jump into as you did?
I think so. I mean, I think there's a ton of problems that are still unsolved, and people are trying to work on them. And obviously, it's something that'll take us decades, right? It's not something that I think will get solved in like a year or two. But at the same time, I think we're sort of at this stage where there's a lot of people working on a lot of different things, and I think it is just a lot of testing that needs to go into validating that these are long-term solutions. And also, like you mentioned, really thinking about like the practical aspect of it. Can we do this at a large scale? Can we manufacture this chemistry at a large scale? And how do we, sell it and really test it in the right way? And so I think generally, a lot of cool work. I definitely think there's more smart people we need working on these problems.
Mm-hmm. Great. All right. So we're going to take off maybe the battery and chemistry hat and put on the biology hat. So another organization that you worked with, which I think is really interesting, is the Chan Zuckerberg Biohub. So it's a nonprofit research organization established in 2016 with a generous $600 million endowment from Priscilla Chan and Mark Zuckerberg of Meta, Facebook fame. They, I think, announced a further $1 billion worth of funding. And they talk about bringing together scientists, engineers, and physicians with a goal of pursuing grand scientific challenges on a 10 to 15-year time horizon. For example, understanding the underlying mechanisms of disease and developing new technologies. They've got a broader mission to cure, prevent, or manage all diseases by the end of the century, I'm assuming this century. And it seems like they are collaborating across, Stanford, UC Berkeley, UCSF, and there's even other hubs, I think, popping up now in other cities. So really interesting organization. It seems to be like part of a crop of several organizations, one being the ARC Institute, which have been sort of generously funded by people who've had success in tech and now have an interest in life science and biology. But maybe just starting with the CZ Biohub, like how did you get involved with them initially?
Yeah, I had actually gotten connected with someone. Well, I was working on a project initially, and I had gotten connected with someone at CZI. They're doing some really cool stuff in like cryo-EM and protein reconstruction, and so looking at sort of like ML methods to help with that. And I ended up attending this like seminar that was happening at CZ Biohub that summer, and they were presenting just their research and what they were working on, and I think that sparked sort of like an initial interest. I was like, hey, this seems like a really cool problem. I really like the team, and it was really collaborative environment. You had, like you said, scientists, engineers, and physicians really working together, and it seemed very seamless, right? It was like this seems like a great place to really learn about all aspects of that, and the problems were great as well. And so I ended up reaching out to Shalameda, who's like the guy who leads the computational team. And so he, him in collaboration with other groups they're doing a lot of work around building really large-scale live imaging microscopy around like, the hardware side, but also building the pipelines and building the algorithms on top of that to help us process that data. And so, that was, we had a project idea and we discussed it, and he was like, hey, this seems like a good fit. And so we ended up collaborating on that.
Wow. Well, one of the things that you collaborated on was a project called DynaCLR. There was a paper that I think came out last fall titled, Contrastive Learning of Cell State Dynamics in Response to Perturbations. In reading that paper, my sort of sense of it is that, ultimately there's a problem that we're trying to solve, which is that when, cells are exposed to perturbations, a la viruses or maybe molecules and so forth, kind of understanding how that process unfolds right now is a very manual, kind of tedious process. It can be error prone if it's done right by individual scientists, depending on whether or not they had their coffee this morning, right, or maybe how they got trained or their facilities. And it seems like you've developed this machine learning model that helps to sort of automate that in some way using some specific techniques in AI or ML. So maybe big picture, am I getting this right in terms of the problem that you're trying to solve here?
Yeah, I would say the high level problem is really, if you think about like therapeutic design overall, it sort of relies on understanding how different perturbations like gene knockouts or drugs are changing the dynamics of single cells. But traditionally, the ways to do that is like you use sort of static imaging techniques, or you have gene expression assays that are developed. But that really provides a more static snapshot of the state that the cell is in. And so if you perturb that cell, really understanding the dynamic changes in terms of the phenotypical impact of that, and also the dynamics is a big area. And so the group that I worked with at CZ Biohub was working, like I said, a lot on live imaging. And what's exciting there is you can collect large volumes of data at multiple different channels. So we were working with 5D data across XYZ, and then across time and different frames. And so there was 5D data we were collecting across different channels. And so if you think about the impact of that, you can look at fluorescence channels where you can have viral proteins or organelle markers that you can track over time, or you can just look at label-free channels that give you a really good understanding of the physical structure of the cell. But the hard piece there is, how do we annotate this data, right? And that's a super manual process that can be very slow and labor intensive. And so the idea there was, can we build a more efficient pipeline to understand that and do some really cool downstream analysis based on those embeddings that we get from these models, to then understand how infections, for example, are impacting the cell, or be able to detect rare events in these cells like cell division.
Well, maybe zooming in a little bit. So the paper talks about DynaCLR being a self-supervised learning framework that leverages contrastive learning specifically adapted for analyzing biological time-lapse data of cells and organelles. Maybe just breaking that down a little bit. So, self-supervised learning, again, in my understanding of it, versus supervised learning. In supervised learning, you kind of have the answer key, right? You tell a model to maybe make some predictions, and you can actually tell whether those predictions are right or wrong based off of some existing, already annotated, accurate data set. In self-supervised learning, you're kind of asking the model to do more of the work and kind of guessing itself. So I think one example they give in the worlds of language is a sentence with a word taken out and has to figure out what that missing word is, or maybe parts of an image that have been sort of removed and has to fill in that image itself. Can you share more about sort of why self-supervised learning was maybe the way to go for what you were trying to achieve versus other approaches?
Yeah, I would say one of the arguments is obviously you don't have a ton of annotated data, right? And so you don't have ground truths, so to say, that you can use to train these models and validate the accuracy. But also really there's an argument to be made about using these embeddings that can sort of learn a more general representation of different cell states. And that was really the argument that we were making in the paper is, hey, if we use a specific training objective function, which in our case was using the triplet loss, which is commonly used as one method in contrastive learning, we can actually really, if we structure that in a clever way and adapt that to these really large scale 5D images that we're collecting, that can actually help us then learn a more general representation of understanding different effects of these perturbations.
So you talked about contrastive learning. Can you maybe share more about how do you define contrastive learning?
Yeah, I would say there's different sort of methods. There's the SimClear paper, which most people have sort of worked on and iterated on. And so we used sort of a variant on top of that, which I call triplet loss. And essentially, I guess the idea there with our framework was we had, let's say, an anchor pair, which could be just let's say, an image of a cell, right? And then that can be a cell at a very specific randomly sampled time point, and then you build a positive pair, which is let's say the same cell but at a later time point. And so at that point you've sort of seen the, let's say, infection progress a little bit further, and you're seeing the impact of that. And then let's say your negative pair is just a completely different cell. And the idea there is within physical space you have these images, and so which images belong to each other and which ones are closer in terms of time, and can we really do a similar parallel mapping within embedding space. And so the way that contrastive learning does that is the goal is to bring together the positive pair closer to the anchor pair within the sort of embedding space, and then the negative pair should be further away from the anchor pair, since that's a different cell. And so using that sort of framework, in our case, that was sort of the argument we were making, and we tried out 3 different sampling techniques that we also describe and run experiments on. One is sort of more of a classical contrastive learning method. We looked at cell-aware sampling. We also looked at cell-aware and time-aware sampling to show how adding that temporal effect can be useful. And a lot of my work was focused on sort of looking at cell division. So we used this open source data set, Alfie, where they have a ton of data and annotated data actually on cell division, and really from the beginning all the way to the end, the cell going through mitosis and whatnot. And so you can really use that as a way to see, is our model learning these rare sort of cell division effects that are really hard to capture because, in terms of data you're collecting, that could be a really short time scale that maybe your microscope isn't capturing. And so that's one area that we found it to be useful. Obviously also looked a ton at just viral infection, because CZ Biohub is doing a lot of stuff with that. So we looked at Zika, dengue virus. So that's one area. Another area, and then third area was also organelle remodeling. And so we looked at early evidence of that within ER remodeling, but also expanding that to other organelles as well.
When you develop these kinds of embeddings, is the idea that once we've develop those, we can generalize across different cell types and different types of viruses, for example, besides dengue or Zika, or are they really specific to the kind of cell lines that you've used in these specific types of viruses?
Yeah, so I would say DynaCLR is more of a framework for what we found in terms of setting up a training pipeline that really gave us interesting results. And I think using that framework, you can definitely build a foundation model. And we do some of this work as well in the extended preprint where we expand to different cell types. And Alfie was one part of that effort because they have a lot of different cell types that they're looking at there. And so initially we looked at A549 cells, which is a specific type of cancerous cell, but you can also expand that into other cell types. And so that's really the argument to be made with embeddings, is that you can generalize to different cell contexts.
So, you mentioned that there's this kind of automated, maybe microscopy component of this that they're working on, and then there's also the, machine learning algorithms that, you were working on and developing. As we look to continue to make progress in this field, I guess, do you find that, more progress is going to be unlocked through just, again, getting access to more data by developing, better and maybe more, kind of these microscopy workflows that are more automated and can generate more data sets? Or is it going to be maybe more on the sort of ML and kind of math side of things?
I think it's definitely a combination of two, right? And I think you need, they sort of go hand in hand. You need a lot more data for these models to eventually scale and for us to be really confident in the predictions that they're making. And so that will definitely require a lot of innovation on the hardware side. But I do think there's a lot that can still be squeezed out from the data we already have on sort of the method side. And so, obviously there's different modalities you can look at. We were looking at microscopy data, but you could also look at, gene expression data, and there's a lot of stuff, happening around just multiomics and using different types of techniques and merging them to draw, greater insights. And so I think there's, the scaling of data, but also just using a multimodal approach that can help unlock a lot more. And then there's sort of a methods side of it, which is just how do we use the data we already have and extract more from it.
Very cool. Well, I imagine that as you are, developing, it's called DynaCLR. Is that how you pronounce? Okay, not DynaCLR. Corrected. So.
I mean, you can say it either way.
Okay. DynaCLR sounds a lot cooler, I think.
Okay.
So, as you were working on it, I imagine, some of the challenges that you might have been dealing with are just, for example, the volume of data and of images, right? And being able to, work with them in ways that are computationally, efficient and so forth. There might be sort of variability and noise within some of the data that you're looking at. So, I don't know, was there a specific sort of, interesting challenge that you were involved in or saw the team, really have to overcome in order to make this work?
Yeah, I worked closely with Zuen and Ed, who are part of the team, and I'd say the biggest part sort of in terms of processing the data in an efficient way was just one, you have terabytes of data. So I think the hardest part is really just how do we get this data to load efficiently. And so I think there is a lot of pipelining work that went into that, so making it just generally more efficient. But also I think there's a lot of stuff within the actual data collection and just the pre-processing that goes into registering the images and making sure that if you go in and it's not like you just collect the data and train these models, right? There's a lot of, I think, human just inspection that also goes into making sure what you're seeing is validated with the actual data. And so predictions our model would make, we actually went in and we would see, hey, looking at specific cells and data frames, are we actually seeing similar results with the work that we did? And one thing that I think helped with that, we had a specific sort of infection sensor that we had built. And so that was really helpful for just having a GFP reporter that would basically translocate into the nucleus. And so if you have, microscopy, you can see that under sort of a fluorescence microscopy and see, hey, the cell is actually infected now. And so we would see, different patterns for infected versus uninfected versus dead cells. And so, even just using the human eye, you could see, hey, what's going on in this cell. I think that was a really useful way to sort of make sure the results we're seeing make sense. But yeah, I would say there's definitely the processing side of it, but then also a ton of just validating the results and actually spending time closely looking at the data.
Well, I'm getting appreciation for the processing side of it. In my conversation with Hani Goodarzi of the ARC Institute, when he was talking about collaborating with Greg Brockman and the team on building Evo 2, I think one of the big challenges was just scaling up to a lot more GPUs. And they're really not even biology problems. They're sort of systems and ML engineering problems that maybe aren't the most exciting, sexiest problems to solve, but you really do need to solve them in order to be able to do interesting analysis at scale.
Yeah, definitely. Yeah.
Great. So speaking of the ARC Institute, so you are recently a research fellow with them, I think starting in the last December, maybe, if I'm not mistaken. And you are working with Yusuf Rouhani, ML group lead, and Hani Goodarzi, who I mentioned, on perturbation modeling. I guess how is that similar or different to the work that you've been describing at CZ Biohub?
Yeah, I would say at ARC, our goal is really to collect one of the largest sort of single cell data sets. And so there's a lot of efforts around data collection methods. And so, collecting chemical genetic perturbation data, just looking at, single cell gene expression data. So there's a lot of effort around that, but then also using that data to sort of build really large foundation models that can help us predict perturbation effects, whether that be genetic perturbation, chemical perturbations. And so, yeah, I would say, that's what a lot of the focus has been on, is using that sort of modality of data and building the architectures around that. And so, yeah, I guess the biggest change is really, I would say the problems are obviously both really important, but the type of data you're using is different. And I think the value in microscopy versus using, gene expression data, I think they both are complementary in my mind, and I think we need sort of a multimodal approach to be able to really understand perturbation effects. And so I think that's what I hope to get from this experience, is more so what does working with this data look like, especially as you, like you said, you scale things up a lot. And so that's a huge effort at ARC, is making sure we really have the largest models and the largest data sets.
Well, I think we touched on this earlier in the conversation, but organizations like the ARC Institute and CZ Biohub, and there are others as well, that are sort of well-funded through these generous endowments, trying to take sort of, it seems like, a new approach to how science is done. You've worked now with a couple of them. I'm curious, what do you think is their role, in this current environment in the future for really advancing, again, AI for biology and for life science? What unique role do they play, or would you like them to play?
Yeah, I think there's a ton of just foundational work that needs to be done, and so I think it provides sort of a non-traditional path of funding, which is really interesting, especially for projects that are at a much larger time scale, right? If you're working on something that might be 10 years out or even longer until you see those results. I think having organizations like ARC or CZ Biohub, it's really useful because they have sort of that long-term mindset. And also, I definitely think it's super mission-driven, right? Having spent time at these organizations, and I think that can be really motivating to just be in that environment where people are really excited and working towards a mission, even if it might be far out and there's a lot of work that still needs to be done. And so I think it's a really nice sort of environment for collaboration because you can bring together people from different backgrounds and provide them the right resources and infrastructure to get that work done, but also for them to even dream up those ideas, right? That might be harder for traditional funding methods to fund.
Interesting. So there's a sense of, I interviewed a woman named Hemi. She talked about patient capital, and it seems that there is maybe a patience to the investments that they have to be willing to invest in things that can take a decade or two to come into fruition, but that ultimately will help move the whole field along.
Mm-hmm. Yeah, definitely.
Great. Well, speaking of the future, you've got a book called AI for Robotics that's coming out. Could you tell us more about your upcoming book? It's going to be published with, I think, A Press and Nature Springer. What inspired you to write it?
Both me and my co-author were really excited about sort of just the field of AI and robotics in general. I think there's a lot of interesting research happening, but we really noticed this gap in the space. If you look at most textbooks, either they cover really deeply classical methods or deep learning and transformers and all the recent sort of hype that we're seeing, or just traditional classical robotics methods. And so we really saw this gap to publish something that merged those areas and talked about really the cutting edge deep learning robotics research that's happening in the space. And so I would say that's how it came to be. Both of us were had spent time looking at these problems and reading these papers and methods, and we wanted to publish something that would really encapsulate all of it together.
So what does that sort of middle ground look like between the existing resources that are out there? I mean, are you talking about sort of embodying maybe new versions of, I don't know, whether it's large language models or machine learning models, and pairing them with new types of actuators? Like, can you give us a little bit more detail about maybe, what the chapters are in your book and what you're teaching us about?
Yeah, we really walk through, I would say the first half is more foundational in terms of just understanding a lot of the key methods. So we talk about perception, for example, and a lot of methods using, all the way from CNNs to using maybe vision language models that are more commonly used today. We walk through, how to think about that paradigm and also how these methods actually work under the hood. We talk about, planning, prediction, tasks, in robotics, what are the recent methods that are going into making those things work. We talk about collecting data in different ways, like imitation learning, reinforcement learning. We talk about using simulations, for example, for data collection, sensors and like how different sensors work. So really covering, I would say, a full ground of how all the key pieces work and then how they fit together. And then the second piece of the book is more focused on applications. And so how do you really take these methods and deploy them into the real world? So we cover, self-driving cars. How do they work? What is the market growth looking like? And where is sort of the biggest impact in that space? And then, industrial robotics. We also talk about consumer robotics and, cover the general market and then also sort of the key methods that are being used in those spaces right now. And so I would say the goal is really to give a good overview of the theoretical aspect of how these methods work, but then also very practically, how do you use those and deploy them into the real world?
So, gosh, I think it was probably a year ago, I attended a talk by somebody from CSAIL. It's an artificial intelligence lab at MIT, who was working in the robotics arm of it for I don't know however many years. And he gave me an appreciation for some of the challenges I didn't think of when it comes to trying to get robots kind of out into the world. So a couple of the ones that I remember just off the top of my head was, for example, if you take anything like a matchbook and you kind of throw it at a desk with what you think is the right force, every single time it's going to land on that desk in a slightly different place, right? Because tiny nuances and friction of, the laminate on the desk versus the thing on the remote, and, the humidity in the air and so forth, makes it so that it's a very hard problem to kind of predict each time. He talked about some of just the cost of like all the different joints on these robots that, can wear out easily or that can break, as you're trying to iterate with them. Which is why he said if you look at the joints on a lot of the robotics, like they're not trying to imitate the human hand. They're a lot more simplistic because they're easier to manufacture and to repair and so forth. Again, at the same time, since then, I've seen a lot of really splashy videos on YouTube of companies that are coming up with these humanoid robots from the US, from China, and so forth. Seems to be like a lot of progress being made. Is this indicative of us really having solved some of those problems in the last year because there's a lot more investment? Do we still have a long ways to go? Like, what's your take on where we're at in that kind of journey?
Yeah, I think everyone has a different perspective on this. I think we definitely made so much progress even since, we started writing the book. I think we had to go back and, writing a book takes a long time. So for us, it was, it took a year or two to even get the initial draft. And once we got to that point, we were like, oh my gosh, there's so much new research coming out. And so we had to go back and actually update some of the chapters to make sure they were really relevant. I think definitely on like the research side, there's a lot of new methods, and I think partially because of sort of this parallel field of just ML and large language models and just compute and data sets getting really scaled up, and so you can leverage sort of these large pre-trained models and the rich knowledge that they have to then build methods on top of it, and I think that's helped move robotics forward as well. But then also just thinking of clever ways to collect data or do teleoperation and whatnot to help improve the methods themselves. I would say definitely a lot of efforts in terms of moving the needle. And on the hardware side as well, right? Like you mentioned, there's a lot that I think can still be done. I'm a big fan of sort of the low cost robotics methods. I think those are, especially for the hardware systems, I think those are really useful and also really foundationally help with research, right? If a lab is able to afford these robots, or even as someone who's a hobbyist, if you can afford these robots, I think there's so much you can do just from your home building these methods. And so I think lowering the barrier of entrance for people being able to work with these robots is one big area that has helped push things forward as well. Obviously still more work to do, and I think it's an exciting field. Yeah.
Well, you mentioned applications in the second part of your book, for example, self-driving cars. I'm curious, given your background and now experience in the wet lab, on the battery and material side of things, what is the future for robotics in science R&D?
Yeah, I think you mentioned the A Lab. I think what they're doing is super cool, and even like when I visited Boston a few months ago, I went to this lab at MIT and got to see sort of their robotic, like very simple synthesizer, but they were working with like powders, which traditionally is a lot harder to work with for robots, and so they were building sort of like the first prototype for that, and I think there's it's all to say there's a lot being done in that space, and I think a lot more labs are starting to do automation, even if it's very simple. How do we just get this robot? I wouldn't say simple, but it's like starting with more repetitive tasks, like pipetting, for example, and automating those and moving towards harder tasks. I think there's a ton of value in looking at automation, and in my mind, I don't see it as like, oh, it's going to completely replace labs and the way that these labs work, but I think it'll will work in humans will be working in tangent with these robots. To save time and really shift their attention towards thinking more about hypothesis and experimentation and interpreting those results, right? And so if there's a way we can speed up the actual repetitive nature part of it, I think that helps unlock more efficiency.
That would be great, to really offload some of those tedious and repetitive tasks so that folks can focus on maybe some of the more creative and interesting parts of their work. Well, I guess maybe just to close out here, I'm curious, as you look into the future, and maybe the answer is it's my book and you should read it and pick it up, but what future trends or emerging sort of technologies are you really excited about, especially when it comes to accelerating scientific discovery?
Yeah, I think it's like a lot of the things we touched on, right? I think automation, using robots and automation in these fields. I think that's a huge area. That was something that I was like really curious about at one point. I remember I looked a lot at like robotic synthesizers, and like I built this like really mini prototype just like in my dorm room because I was really excited about that space, and I think there's a lot that can be done there. Yeah, I think generally like building sort of like these foundational single cell models, which is the work that I'm working on now, and work that I did with DynaCLR is another big area that I still think is very, very much like underexplored, and there's a lot more that can still be done. It's a problem that we've been trying to work on for a long time, of course, but I think there's we've reached a point where there's more data, more compute, and so thinking about it from a different angle, I think is exciting. I think there's a lot of really cool stuff in like protein language models, structure prediction. That's an area obviously that AlphaFold and other methods have been working on, ESM, and so I think if there's ways we can really push that forward, and a lot more that can be done there in terms of like using like these genome language models and DNA. And adding that into these models, I think that can unlock a lot for not even just understanding, proteins, but potentially modeling the entire like transcriptomics, and transcriptomic data can be really interesting as well.
Well, one of the things I appreciate and love is the way in which you have played around in really interesting applications of, again, machine learning to different areas of science, and now moving into robotics. I'm curious, as somebody who might be listening to this episode who is themselves in high school, or maybe themselves mid or late career, and is intrigued by these kinds of technologies, but maybe a little bit intimidated by them, kind of not sure where to start. What advice would you have for them in terms of how to get going?
Yeah, I mean, I think there's a lot of value in just like giving it a try, getting started. I think that's sort of the mindset I had, right, as I mentioned, starting out just in high school and attending these like AI talks in Toronto. That's sort of how I felt at that time, right? When you speak to people who have spent decades in this industry, it almost feels like, okay, where do I get started? Where can I be the most impactful in terms of my skill sets? And so I think just getting started and starting to even just self-learn, there's a ton of value in that. And obviously, taking classes at school or shadowing people who have been working in the space, I think is another big thing. I think you'd be shocked how often people are just willing to, chat and help you out. And so taking that first leap of faith and reaching out, whether it's like a professor at your university and you really like his course or her course and you want to chat with them about it, or a lab that you might want to join. I think that's another great way to learn and get your hands dirty, especially with the science. I think those are like two areas that are more accessible perhaps, but also just starting with resources that are out there online. I think that's the exciting thing about the time that we're in. If you just have access to a computer, there's so much you can do with just that. And so, spending time, I think learning and reaching out is really useful.
So just get started, just ask, and you might be surprised, how far you can go with just those two things.
Yeah.
So, we've got folks who are listening to this who might be, investors, policymakers, people in industry and so forth. One question I always like to ask is, how can people be helpful to you and to what you're working on? So I'll ask that to you as well. How can we be helpful?
Yeah, I mean, I think obviously I think with the research I'm doing, I'm always happy to, chat ideas or just chat about generally what I'm working on. And if people have, ideas or want to collaborate on things, I'm always open to that. I guess other things I will mention, for the work we did with DynaCLR, we also have a second preprint coming out soon. And I think if you are interested in more of sort of the biological aspects of how this can be useful, that will have a lot more results that the team has been working on there. And so highly recommend checking that out if you're interested. And then, in terms of like the book, I think if you want to learn more about sort of the methods behind a lot of these algorithms and how things can be applied to, physical hardware, robotics, checking that out, I think would be another area that could be great. But I would say, those are the main things. I mean, I'm always happy to chat about ideas and research as well. Okay, beautiful. If folks want to get a hold of you, what's the best way to do that? Yeah, I would say Twitter is a great way. So it's just my full name, _ and then, I'd say Twitter, LinkedIn, or email as well.
Beautiful. And then if folks want to pick up your book, where do they do that?
Yes, it's on all the major sites. I mean, you can check out Amazon. I would say that's the biggest place that you can buy it. And then also the Springer Nature website has a online version as well as a physical copy that you can get.
Okay, awesome. Well, Alishba, thank you so much for coming in and spending this time with me and with us today. I'm personally just really inspired with the bold and entrepreneurial way in which you have really explored the intersection of AI and science. I know it's inspiring to me personally, so I appreciate your story and excited to see the cool career and projects that you are carving out next.
Yeah, thanks for having me.
Absolutely. All right. Well, thank you for joining this episode of Discovery Engines. If you want to learn more about some of the organizations and resources that we've mentioned, check out the notes in the episode comments. And otherwise, you can also subscribe to our newsletter at discoveryengines.co. See you next time.